What's up, distinguished boys and girls? Welcome back to my channel. The PDF that I told you about that I was working on is ready now. I'll link it in the comments. I think you should go and check it. It's very solid. I gave all the possible questions that you can get. At the end, I provided also some questions from the previous years, and I translated them to English. So you'll be missing out. And I'm providing it for free. And now we'll just continue reviewing the topics. So if you get a question telling you to mention the notable differences between blue-green algae and bacteria, or explain or justify that bacteria and blue-green algae have some differences even though they belong to the same kingdom, the answer here will be because bacteria have no chlorophyll pigment. Bacterial cell wall is surrounded by capsule, while blue-green algae cell wall is surrounded by gelatin membrane. This here was in the previous question copies where they put the gelatin membrane as a blank question. Also, you want to pay attention to the drawings. You don't want to miss them. I think every drawing question is just a free question. If they tell you explain or just explain, eukaryotic cells have varying shapes, or eukaryotic cells change shape from time to time like amoeba. Because they change in shape due to the function of these cells. Okay, so cells often have shapes that adapt their function. Okay, so the shape changes according to the function of that cells. Amoeba here means any cell that is able to change its shape. Basically, a cell that is able to put some processes we call lamellipodia. Usually, cells that perform phagocytosis. So those lamellipodia helps in cell movement. Okay. So, as I said, usually in cells that perform phagocytosis, like the white blood cells, which are gonna help the cell to move. So amoeba here is just a generic term. Any cell that is able to do that, we call amoebic feature. But this book refers to amoeba as if it was a separate organism, even though it's not. Amoeba can mean any cell that is able to perform this feature of changing shape. Questions like give the function for each of the following is very common. They will tell you give the function of the plasma membrane, for example. You will just write down that it exchanges materials with surrounding environment appropriately, marks the outer border of the cell, support and protect the cell, and maintain the liquids inside and outside of the cell, and blocks large particles to diffuse across the cell, because it is selectively permeable. Another possible question style you can get from this is give an example for each of the following, and they will tell you a cell component or a cell organelle that marks the outer border of the cell, and the answer will be plasma membrane. If they tell you what are the major components of the eukaryotic cell, very important, it's cell wall in plant cells only, and plasma membrane in both plant cell and animal cell. Plasma membrane and cell membrane mean the same thing. They're just synonyms to the same structure. And of course, we will have the cytoplasm and the nucleus. Those are the major components. Also, what is the function for each of the following? The cell wall. The function is protection and support to plasma membrane and the cytoplasm. Okay? The cell wall in plants is made up of 3 layers. You have to memorize this by heart, as you can have this either in a blank question or in the definition question where you're going to have to mention everything you know. So the 3 layers are middle lamella, primary wall, and secondary wall. As you can see this in the picture here, or in the image here, we have the primary cell wall. This the superficial layer in green, and the secondary wall is this deeper layer in blue. And the middle lamella is this one here in orange. And this is basically to the outside of the primary wall. And it connects the primary wall of a cell to another primary wall of another cell. I'm providing this picture so that the memorization process is easier for you. Visuals always help in the memorization process, so that's why I'm providing this picture. If they ask you about the origin of the middle lamella, this one here, it is originating from the cell plate during cell division. So during cell division, plant cells have something called the cell plate originating from the protoplasm or the spindle. Okay, so again, this is very important. During cell division of plant cells only, we have something called the protoplasm. Protoplasm means the cytoplasm plus the nucleus plus organelles. Okay, so it's a term that referring to a lot of things. Okay, another possible answer or another correct answer you can write instead of protoplasm is the spindle. Okay, so it's originating either from the protoplasm or you can write down the spindle. It will give rise to the cell plate during cell division. The cell plate is the structure that will help make up the new cell wall of the new divided cells. And it will technically give rise to the middle lamella as well. Also possible question, give the chemical composition for each of the following and the cell wall in plant. It's just cellulose in young cells and lignin in older cells. If you get a question being specific about either plant cell or bacterial cell, for example, cell wall in bacterial cell, we will just say that it is made up of proteins, lipids, and polysaccharides. But if the question was not specific and they just want the chemical composition of a cell wall without being specific, so you will write down either the chemical composition of the cell wall of the plant cell or the chemical composition of the bacterial cell. Both answers will be correct at this point. Of course, the drawings, you don't want to miss them. We will discuss the plasma membrane real quick. So the plasma membrane, you want to memorize this by heart. It's very important. So the plasma membrane is found in both prokaryotic and eukaryotic cells. It is flexible. We will write this in the comparison question between the plasma membrane and the cell wall. We will write down it's flexible, comparing it to rigid / non-flexible structure in the cell wall. Okay, so it's flexible, it's thin, and the cell wall will not be thin, it will be thick. Where is it? Here. So it's thin membrane. The cell wall will be thick membrane. And of course, you want to mention that we cannot see this by light microscopy because it's very thin. Instead, we will use the electron microscopy. The cell wall, on the other hand, we are able to see it just using light microscopy because it's very thick. Chemically, so the chemical composition of the plasma membrane is 2 layers of phospholipids. As you can see, this is one layer, and this is the second layer. Phospholipids are made up of hydrophilic head, meaning its water-loving part, and hydrophobic tail, which does not like water, hates water, afraid of water. You can see that the hydrophilic head faces the outside and the inside of the cell where water is found. And on the opposite side, we will have the hydrophobic tails. Between those phospholipid layer, we have protein particles. The function of those proteins, they control the movement of material, okay? They control the passage of materials. I will try to give you the big picture here as why this is controlling the passage of materials. So basically proteins form something we call protein channels. The protein channels basically control the movement of materials, as I said. For example, the simplest example I can give you is the aquaporins, which are the water channels that control the movements of water inside and outside of the cell. If they ask you about the location of the plasma membrane, you will just write down that it surrounds the cytoplasm of both eukaryotic and prokaryotic cells. Okay, so if they ask you about the cytoplasm, and the very possible question is, what is the location for each of the following? And they will tell you the cytoplasm. You will just write down that it lies between the plasma membrane and the nucleus. Now the cytoplasm, let's go back to the picture. As you can see, this is the cell structure here. The outer border is the cell membrane that we discussed. And this is clearly the nucleus and the nucleolus inside of it. Everything here you see in yellow or whatever this color is the cytoplasm. So the cytoplasm is the fluid or the fluid-like substance that is found throughout the cell that other organelles float on. Okay? So you can imagine it like the fluid that other organelles bathe on. Okay? So everything here is the cytoplasm. So when we want to describe the location of the structure, we will just say that it is from the cell membrane here to the nucleus here. So everything from the cell membrane to the nucleus is the cytoplasm. This is the location of it. Okay? One more location, the other location that is in common with the cytoplasm is the endoplasmic reticulum, as you can see here. This is the endoplasmic reticulum. When we want to mention the location of the endoplasmic reticulum, we will also write down that it is between the cell membrane and the nuclear membrane. Okay, as you can see it here. This is clearly the rough endoplasmic reticulum, as I said, because it has the ribosomes, it looks granular, and this is the smooth. It does not have any ribosomes. Okay. Another question you can get is the chemical composition for each of the following, and they will tell you the cytoplasm. It's just 80% water. Remember that water makes a major component of the cytoplasm. 15% protein and 5% fat, sugar, and salts. You will have to memorize those proportions even though they can differ from one cell to another. It's not necessarily 100% correct. Though this question was present in the previous copies, they put this as a blank question. They like the chemical composition of the plasma of the cytoplasm, and this was a blank, the 80% water. Also, another blank question you can get is the organelles. For example, we have living structures in the cytoplasm, which are the other organelles, and the non-living components, okay, which are particles formed by the activities of the organelles. The non-living components can be lipid droplets, enzymes, hormones, pigments like in melanin, and they are all formed by the activities of other organelles, for example, in cell metabolism. If they give you a question where they're going to say, give an example for each of the following, and they will tell you a living component of the cytoplasm. Here you can mention any organelle you know. endoplasmic reticulum, Golgi apparatus, mitochondria, plastids, vacuoles, kinetosome, etc. Anything will be correct. The non-living components here, they will either tell you to mention everything you know about it, which are the 5 items here, or they can give you this question style, give an example for each of the following, a non-living component, for example in the liver cell. And the answer will be carbohydrate accumulation, represented by glycogen. Also, a possible question are the blank questions. You want to pay attention to that. This point here is important because they mentioned something about the neurosecretory granules. There was a question telling you to mention the characteristic or the feature of the neurosecretory granules, and the answer will be granular, spherical, or oval, surrounded by a membrane. This factoid is mentioned in this point, so I think this question is very weird and very specific. That's why you have to pay attention to every line. Alright, so the endoplasmic reticulum, we discussed the location already. We will talk about a question you can get: why is it called endoplasmic? Why is endoplasmic reticulum is called so? Because it is a reticulate, interconnected system of tubules and vesicles that interlocks and branches with each other. It's very possible that you might get a question like that. Why is endoplasmic reticulum called endoplasmic? This is the reason here. The function of it, it's basically the place where lipids, carbohydrates, and proteins are made. For example, in rough endoplasmic reticulum, as we said, why is it called rough? Because of the presence of the ribosomes, which give it a rough and granular appearance. The function of rough endoplasmic reticulum is active role in building proteins. Now, in order to remember that, you can just imagine, or you can just imagine that the ribosomes are the main components of the cell that make up the proteins. So, the presence of ribosomes here make the rough endoplasmic reticulum also being able to build proteins. And the other 2 functions are exactly the same mentioned in the smooth endoplasmic reticulum. They're in common here, so that's very easy to memorize the 2 points here, because they're in common with the smooth endoplasmic reticulum, which are basically help transport material inside the cell, especially to Golgi bodies. Now, Golgi bodies here means Golgi apparatus. Okay, it's the same structure. So if they mention Golgi bodies, or Golgi apparatus, or Golgi complex, all of those are synonyms to the same structure. The other function is that it acts as a structural reticulum of the intracytoplasmic material. Okay, so it's structural reticulum. It's basically structural support, a network for the intracytoplasmic material. The smooth endoplasmic reticulum are called so because, as I said, their surface do not contain ribosomes, so they are smooth. The function here, again, we have 2 functions are in common. Those, we mentioned them. And the other functions are very easy to remember. They just play a role in detoxification of drugs and medication. This is very important, as you can have a question, give an example for each of the following, or who's responsible for each of the following. And they will tell you detoxification of drugs and medications, and the answer will be smooth endoplasmic reticulum. The other functions are they build and concentrate fats, and they secrete the steroid hormones. Now, question you can get: Why is smooth endoplasmic reticulum is abundant in the cells of the ovaries, testicles, and the adrenal glands? Because it builds and concentrates fats. And we know we need those fats in order to secrete steroid hormones. Okay? Steroid hormones basically means the testosterone in the testicles, the estrogen and the progesterone in ovaries, and the aldosterone, cortisol, and other androgens in the adrenal glands. Okay? Also, you can get a very important question to compare the smooth endoplasmic and rough endoplasmic reticulum. We discussed everything, so I'll leave that to you. We can have a lot of questions in Golgi apparatus. Who discovered it, the name of the scientist, the year that he discovered it, and how did he discover it while he was studying neurons. So here we can get blank questions. The Golgi apparatus location, it has a special location in the cytoplasm between the nucleus and the plasma membrane. So this is the third component that have the same phrase. It's between the nucleus and the plasma membrane. But you will have to continue writing that it is hard to identify the certain location and it differs from one cell to another. One more thing that can make students panic is the dictyosome. Dictyosome is basically the Golgi apparatus, but in plant cells. That's it. And its function is to build cellulose and some other components of the cell wall. So if they give you a definition or the location or the function of dictyosome, if the location it's the same as Golgi apparatus, there's nothing going to change. Okay? Definition, you will just have to write whatever you know about it. And the function here, you're going to be specific because it's only in plant cells. Okay, cellulose. So dictyosome basically is Golgi apparatus in plant cells. There was one question. They just said that Golgi apparatus has no, and then blank question. Okay, so Golgi apparatus has no and blank. The answer is ribosome. It's very weird. So Golgi apparatus consists of 3 chambers marked by smooth membranes. Okay, so in Golgi apparatus, we have 3 chambers and separated by smooth membranes. The first one is cisternae. This is, usually you're going to have this in definition question, define the cisternae, and you're just going to write down that it is the first chamber of the Golgi apparatus made up of flat 3 to 10 saccules. The second chamber is vesicles, made up of vesicles, and the third chamber made up of large vacuoles. You can also get blank questions here. Or they will tell you a question to describe the structure of the Golgi apparatus, and you're going to write down all of that stuff. Golgi apparatus function in animal cells is different from the dictyosome in plant cells. Because here we will have building and secreting complex sugars, so it is able to build and secrete the sugars. It secretes proteins only. It does not build proteins, okay? It just secretes proteins. This protein is obtained from the endoplasmic reticulum, as we discussed earlier. We said that endoplasmic reticulum transport materials to the Golgi apparatus, and one of those are the proteins. Okay? So if they ask you about why Golgi apparatus only secretes but does not build proteins, it's because it lacks ribosomes, so it does not have ribosomes to build proteins, and they only secrete it because they obtain it from the endoplasmic reticulum, which is able to build proteins due to the presence of ribosomes. The other function is that it secretes many materials like hormones and enzymes. That's it. Right. So, the mitochondria here, if they ask about the shape, it can be either spherical or filamentous, as you can see here, and differs according to the type of the cell. You'll just read this yourself. I just want to mention the most important things in my video. The cristae are the curves and folds of different shapes in the inner layer of the double membrane of the mitochondria. So here, you can see the mitochondria. It is surrounded by a double membrane, as you can see here. The inner membrane has a lot of curves and folds, and we call them the cristae. The function of those cristae is to increase the surface area of the inner membrane of the mitochondria. Okay? Do not confuse cristae with cisternae. Cisternae are found in the Golgi apparatus, the first chamber, and cristae are the inner layer, or the folds and curves of the inner layer of the mitochondria. And the function is to increase the surface area of the inner layer. They will tell you, explain or justify why is mitochondria known as the energy house or the powerhouse of the cell. Because they are involved in producing the high energy ATP, or adenosine triphosphate. Another justified question is the basic function of mitochondria is cellular respiration. It's because they produce respiratory enzymes. Okay, so we have two functions here. The plastids found only in plant cells, so it's not in animal cells. So if you want to compare the animal cell with the plant cell, you will write down that the plastids are found in plant cells but not in animal cells. We have three types of plastids. We have the chromoplast, the leucoplast, and the chloroplast. The chromoplast will give the different pigments and colors to the flowers and fruits. The leucoplasts are the colorless plastids. Okay, their function is to convert glucose sugar into polysaccharide, like starch, or into fats or proteins. Okay, so glucose sugar is the simple sugar that we can get, and I'm going to convert it to polysaccharide, meaning a longer chain of sugar. Okay, so it's sugar converting to a longer chain of sugar, like starch. Starch is a very long chain of sugar. Or I'm just going to convert the glucose into fats and proteins. This is the function of the leucoplast. They can also give you a question, who's responsible for each of the following? And they will tell you converting glucose into polysaccharide. And the answer is leucoplast. The white matter in potatoes, for example, result from the colorless plastids and is full of starch. Here is also a question telling you who's responsible for each of the following, and the white matter in potatoes, the answer is leucoplast, which is full of starch. Okay. Remember that leucoplasts are colorless. And finally, the chloroplast. It is surrounded by a double membrane. And the chloroplast is the plastid that contribute in photosynthesis. So we have on the inside of this membrane, so here we have our structure. This is the outer membrane, and this is the inner membrane here. Okay, and we have intermembrane space. In the inner membrane, we have something called the thylakoid. Okay, so a single disc or a single oval that you can see here is called the thylakoid. And the stack of the thylakoids, for example, we take those 3 stacks here, or the stack made up of 3 thylakoids, we call it granum. Okay, so granum is basically the same as thylakoid, but the thylakoid that are in stack together. Okay, so stack of thylakoids is called granum or grana. Those grana are connected to each other by a lamella. Okay, and this fluid here is called the stroma. It's aqueous fluid. Okay, so all of this is to the inside of the inner membrane. So the stroma again is the liquid transparent material. Also, you can give a question. You can have a question telling you to give an example for each of the following, and the liquid transparent material that is to the inside of the inner membrane of the chloroplast, and the answer is stroma. So the stroma contains the grana, as you can see here. We said this is all of this is the stroma. It resembles the cytoplasm. Okay. So all of this is the stroma. And to the inside of the stroma, it contains the grana, or the thylakoid discs. In the stroma, we have the CO2, which is a reducing enzyme that helps chloroplasts with photosynthesis. And in the grana contains the chlorophyll, which is the pigment that helps in photosynthesis. Thylakoid, you can have the thylakoid as a definition question. It's a very common definition question. You're just going to say that it is a capsular structure formed by the inner membrane of the plastid. Contains chlorophyll, the pigment, and enzymes which help in photosynthesis. Okay? So again, the chlorophyll, we can say that it is contained within the thylakoid or within the grana because they are technically the same structure. If they give you, this is very important, if they give you a question and tell you, give an example of an organelle that is surrounded by a single layer or monolayer membrane, the answer is lysosomes. Okay, so lysosomes is surrounded by a single membrane, a monolayer membrane, single layered membrane. Okay. So the lysosomes contain enzymes that help in digestion. For example, they are found in abundance in neutrophils. Why? Because neutrophils perform the phagocytosis. For example, during infections, neutrophils are activated to kill the microbes in our bodies. And in order to kill them, we need a lot of lysosomes that will secrete the enzymes that will kill the microorganisms. Okay? This is also one of the questions that you can get, as in why neutrophils is full of lysosomes. One more thing I want to mention is the metamorphosis. You can have the metamorphosis as a definition question, or who's responsible for each of the following, and they will tell you metamorphosis. Metamorphosis is basically when the animal changes shape from one phase to another. For example, in the frog larvae, the tail disappears when they become adult frogs. This is also a possible question. Give an example for each of the following. Metamorphosis, you will write down the frog. Like the frog larvae turning into adults, and the tail will disappear. And this is done. Who's responsible for metamorphosis? The lysosomes, okay? the enzymes of the lysosomes. Who's responsible for each of the following? Recycle elements in nature. Very, very common question. It's lysosomes. The cytoskeleton. So the cytoskeleton, only two components are mentioned, the microfilaments and microtubules. You can have them as a comparison question. Microtubules made up of tubulin proteins, and the microfilaments are made up of actin myosin proteins. The function is, let's talk about location actually. The location is usually in muscular cells, okay, because they're going to help the contraction of the muscles, okay? We can move our muscles due to the contractions that is performed by the microfilaments, the actin and myosin proteins. Meanwhile, microtubules location is actually found in animal cells only. It's not in plant cells. It's only in animal cells and primitive organisms like the algae and fungi. Usually near the nucleus. Please mention this, near the nucleus, very important. Okay, and please mention that it is not found in the plant cells. And the microtubules will eventually form the centrosomes. Okay, the centrosomes that is also found near the nucleus will help the movement of chromosomes towards the opposite poles during the cell division. And it is made up of microtubules. Okay? The function here, form the centrosome, important for the cytoskeleton, organization and transfer of materials, and chromosome movements in cell division. They also are a major element in the formation of cilia and flagella. The shape is tubular, and this is just straight, thin. Of course, microtubules are larger than microfilaments. So give an example of a eukaryotic cell that lacks the centrosome. It's clearly the plant cells. Instead, they have something we describe it as a center for creating microtubules and microfilaments. So here are the structure of the centrosome. We have two centrioles. This is 1 and this is 2. And they are made up of microtubule triplets. We have 9 triplets of microtubule, as you can see from 1 to 9. Triplets means like 3 groups of microtubules and repeated 9 times, okay? So this is the basic structure. Okay, again, I'm gonna emphasize on this one. Centrosomes are made up of 2 centrioles, 1 and 2. And each centriole is made up of 9 microtubule triplets. The kinetosome, or the basal body, so you can have this as a definition. The kinetosome basically located at the base of flagella and cilia, which contain cilia and in cells that contain the cilia and flagella. But the function also has a vital role in the movement of cilia and flagella. Very important. You always have, there are like tons of questions here just talking about the location and function of the basal body or the kinetosome. This is the last thing here, the vacuoles. So they will tell you why Partista, like amoeba and paramecium, have contractile vacuoles. The answer is to drain the cell from the surplus water and dissolved waste, okay? So it's just to drain the cell. We can also have something like temporary food vacuoles. Also, you want to know that cell sap, which is just a juice of different dissolved materials that serve as storage of materials and support the cell, which is found in the vacuoles of plant cells. So if they ask you about the location of the cell sap, it is in the vacuoles of plant cells. If they ask you about the function of the cell sap, you're just going to say that it contains, or it is made up of different dissolved materials which serve as a storage and support to the cell. And the three types of vacuoles are basically contractile vacuoles, temporary food vacuoles, and storage vacuoles. So, what should you know about the vacuoles? Again, they are just structures there in the cell. Their function is to support and storage. Okay. For example, in temporary food vacuoles. And another function, the contractile vacuole, is to drain the cell from water. That's it for now. If you like the video, please subscribe to my channel, share with your friends, and thanks for watching.